We have come together in support of this resolution. It's kind of a catch-22 with awareness. You're trying to raise awareness because no one knows about it, but in order to get people to know about it, it has to be more public. And so it's been a long journey just getting our DIPG community and childhood cancer community to know that it's there. My son Jack was diagnosed with DIPG in 2011 in October. The doctor's eyes were teared and they had a hard time looking at me. They wouldn't tell me exactly what the results were from the MRI, but it was bad. And they notified me, they called me back up into the hospital room where he was still in the MRI, and they said, you're gonna have a whole new team. They were the neuro team that we brought him in to see because he was having neurological symptoms, and thank God I was able to get him into the pediatrician who was hip to what's going on and said, oh, if this were my son, I would take him to the Children's Hospital right now. So we did. But thus began a long journey for us of extreme pain, a lot of joy in knowing the preciousness of life, which is one of the real blessings of this journey, as difficult as it is. But the one thing that has inspired me the most to advocacy for pediatric brain cancer in particular and childhood cancer is the fact that when I asked, why don't we have anything? And the only answer I could get was from a friend of mine. Her husband works for a pharmaceutical company. He said, well, that's just one of those sad, rare orphan diseases, and the numbers aren't great enough for investors. And since that time of learning that, at the time I thought that, it was so rare, I'd been struck by lightning, and, as a parent, you go through all sorts of gyrations in your mind trying to figure out what you did to cause it. Somehow you had to, of course. And it's a mentally trying time, it's a spiritually trying time. And no parent should hear that there are no solutions for their children because the numbers aren't great enough for investors in the wealthiest country in the world, where we pride ourselves on our humanitarian values and the way we value our conservative family values of our children. And to think that we couldn't give this the time of day is unconscionable to me. But people just don't know. People don't know that brain cancer is the #1 killer of children with cancer. That the huge success we've had increasing the survival rates for childhood with cancer is because of all the work we've done, mostly with leukemia. And 40 years ago it was a death sentence. Today it isn't because we targeted it. We called it out by name. And look where we are today. But sadly, despite the increase of survival rates overall for cancer for children, these cancers, the pediatric brain tumors, have not seen that kind of progress. In fact, there's been no, as you know probably just as well as I do, there's no change in the standard treatment protocol of radiation and palliative care. Go take your child, make them comfortable. Since Neil Armstrong's daughter died of this disease in 1962. So this is our moonshot for kids, because we wanted to go as a society to be able to reach the moon. And with 1960s technology, we wanted it so bad, we put them there and brought them back safely. Thank God, probably by the skin of our teeth, right? But we can do this, but we have to be resolved. And that's what the resolution is about. So I am just out of this world grateful to these amazing doctors and Josh Allen with his company Orthopedics, and we have, but the star of the show for me is Jace Ford, who is a pre-law student at Kansas State, and he is willing and able to be the voice for DIPG, because as so many of you know, most of the children are very young and aren't with us as long as we would like. But he is doing super well on UNC 201, and I'm just thrilled that he's with us today to have this opportunity to speak to you. And so our first panelist, and I'm just in so much gratitude to her, she was the original confirmed expert on the first ever DIPG awareness resolution in the state of California in 2014. And she has been my hero ever since. Now she's working on an incredible program in Zurich, and I can't wait to hear more about it. Thank you so much, Sabine, for being here.
Thank you very much, Janet. Really all the credit goes to you and all the families in this room, really helping us in the fight against DIPG and brain cancers in general. So it is really a true honor to be here today and support Janet and all the other advocacy groups to really create more awareness for children and families whose life has been changed forever by being told your daughter, your son has a brain tumor. As Janet already mentioned, and as many of you are already aware, today more children die from brain tumors compared to any other childhood cancer. But unfortunately, support for research for pediatric brain tumors remains extremely limited from federal funding agency as well as of industry, because there are luckily not that many children affected, so it's not a good market for industry. Amongst pediatric brain tumors, diffuse midline gliomas, including diffuse intrinsic pontine glioma that we often refer to as DIPG, are really the deadliest tumors of childhood, and there are virtually no long-term survivors. Children often come to attention at a very young age after only a few weeks of symptoms. Common descriptions we hear from families is, she was just a little bit more unsteady. His eyes didn't move correctly anymore. She couldn't use the left arm. Sooner or later, then a picture is taken of the brain, which unfortunately leaves little doubt about the diagnosis. When we then, as neuro-oncologists, meet with the families for the first time, we not only have to tell them that as a community we have not been able to find an effective therapy for these tumors, but that the time that a family has left with their daughter or their son is extremely limited. The median survival for these tumors to date remains 9 to 12 months, for many less than a school year. And I think that is really important. Less than a school year, the families have left with their children. And as Janet already pointed out, numbers have not really changed over the last few decades. And the desperation of families trying to save their child and our lack as a medical community to offer better therapies for these deadliest tumors have driven families to seek care outside of the US as well as Europe, often putting families in desperate financial situations as well as also emotional situations with very unclear benefit. But I think there's really hope. I remain optimistic that with increasing awareness, the right financial support, and true collaboration amongst key stakeholders such as family, advocacy groups, regulatory agencies, and the medical community, we will be able to change the outcome for these tumors. One big dilemma our field suffered, and I think really limited our success, was that we can't get access to the tumor tissue easily for these tumors. These tumors are located in midbrain structures of the brain that control critical functions such as breathing, the heart rate, and grow really intertwined with normal brain cells. So often a surgical resection is impossible, and for the longest time, even taking a small piece of a tumor was considered to be too dangerous. And then the field unfortunately got caught up in a circular argument saying, we cannot put a child through a biopsy because we don't know if there's any benefit for the child. But obviously, if we never start taking tumor tissue, we will never know if there will be a benefit. So really, through a lot of advocacy from families and a true collaborative efforts, that luckily has changed. And now biopsies are really part of routine care and widely accepted, especially in part of clinical trials. And we have learned so much of these studies, and we learned that taking a piece of the tissue is safe, and really that led to an explosion in our field understanding the true biology of these tumors. And I'm just highlighting this because through this collaboration, through this advocacy from families, we really changed the field tremendously, and I think we can do the same, and we have to do the same going forward. With the collection of tumor tissue, we have seen that these tumors, even if they present at similar ages, that they look the same on imaging, but they have different changes. So what we realized that we cannot design therapies like a one-size-fits-all approach. Each child deserves their own assessment of their tumor and a dedicated therapy plan depending on the specific changes their tumor carries. And so also taking tumor tissue has allowed us to develop animal models that we can use for new drug testing. We are still waiting for all of these great discoveries over the last 7 years that they impact the clinical outcome, but really I have no doubt that as we understand the biology, we will come up with better therapies. But as a community, we have to continue to work together and advocate with families and advocate for these families and children affected by this terrible disease. It always starts with awareness, and this is why this resolution is so critical. Increasing awareness will hopefully also to more groups working together on some of the critical aspects that limits us. For example, opening of trials testing new and promising agents takes way too long, especially when thinking about international trials and combination therapies. For the rare disease like diffuse midline gliomas, agencies from different countries need to work much closer together and allow for a more seamless regulatory approval process. The field needs more industry support, and we will obviously hear from Josh Allen from Oncocytics how that we can move this forward. Data sharing and collection remains a key limitation, and Dr. Resnick will address this further in his presentation. But the desperation of families leads to the expenditure of sometimes several $100,000 of US for experimental therapies per family that sometimes does not contribute really to the larger knowledge in the field since data is not being collected and shared appropriately. But one other key issue remains funding. Most funding for DIPG research comes from dedicated family foundations who are present, actually many of them here today in the room. Our dedicated clinical trial consortium to develop new therapies for children with brain tumors, for example, the Pacific Pediatric Oncology Consortium is slowly philanthropy funded, and so is really the DIPG Center in Zurich, aiming to develop better therapies. And that obviously always leaves us with an uncertainty, do we have enough funding the next year to continue this really important research to improve the outcome for our children. I'm hoping that with all your support and commitment to this cause, there will be a day in the near future when I can say to families, this used to be an incurable disease, but through true collaboration, we now have effective therapies. So thank you very much for supporting this important resolution.
Thank you, Dr. Miller. And now Dr. Resnick from Children's Hospital of Philadelphia, CPTC.
So, as Sabine mentioned, I think for people like us, we are tremendously grateful to be invited on behalf of our community to represent some of these efforts here today. I have to apologize a bit because I might be a little bit haggard because I actually just landed this morning on a trip from Australia. That's a 22-hour flight. I actually flew into Australia on Tuesday, spent 36 hours there, and flew back. And the reason I did that is because in Australia, I think there's a representation of the challenges we face and the opportunities at hand. And so I thought maybe I'll start in that context. Australia is about the size of the continental United States, but they have about 1/13 the population. So each year in Australia, they have approximately 900 cases of pediatric cancers. That works out to about 15 to 20 DIPG cases a year in Australia. So you can imagine that a researcher like myself, I did my training just up the road at Hopkins, and now I'm at CHOP where we have large volumes and collaborating with consortia, as Sabina mentioned. You can imagine that a researcher or a family in a community like Australia might be challenged, right, to really fully harness the opportunity landscape on behalf of advancing research. Because we only have 20 cases a year across an entire country the size of the United States. How do we advance the field? But the other side of the coin is that these types of challenges also offer new opportunities to rethink the way that research is done. And I think that's one of the sort of lessons that DIPG provides, because it provides a context and a saliency around which we can develop new ways of doing research. And so the reason I was in Australia for 36 hours is because we were launching a beta version of a platform called Cavatica, it was Cavatica Australia, to support the capacity of researchers in Australia to connect to and harness data and information generated in the United States and across Europe, and on behalf of patients in Australia. So connectivity across resources that are global are new technological opportunities that change the way we need to do research. Now, Cavatica was actually initially launched here in the United States in 2016 as part of the initial cancer moonshot efforts under the Biden Cancer Moonshot. But it was launched for the very same reason that it was launched in Australia. It was launched in order to harness the opportunity to connect and share and collaborate across data on behalf of accelerated translation. It always struck me, right, partly because I'm an aviation and space enthusiast, that the original moonshot had something like 400,000 people dedicated to the moonshot itself. It had something like 20,000 universities and companies dedicated to it. And so even as we say the word moonshot, it's clear that that's a challenging context to implement on behalf of pediatric cancers or even on DIPG. We don't even have 400 pediatric hospitals across the United States. So we couldn't even have 1,000 investigators dedicated, right, to a single initiative across the country. And so numbers are the challenge that we face, but also the opportunity. So you have to really rethink the way you do research as a researcher who's dedicated to pediatric cancer, and particularly pediatric brain tumors. So as Sabina mentioned, consortia provide a really unique opportunity to rethink this model. This is something that both the Pacific Pediatric Neuro-Oncology Consortium and the Children's Brain Tumor Tissue Consortium early on committed to, which really is accelerated connectivity of information and harnessing expertise across more than 20 institutions in the United States, in Europe, Australia, and Asia. And the key is to harness both the connectivity and willingness of institutions to partner technologies that embrace that capacity to share data both amongst those institutions, but then even globally, but also new practices around the speed at which information is shared. A key thing about the moonshot, the original moonshot, that most people forget is that the reason that it launched was because of competitive environments between the US and Russia. So the reason everything scaled and accelerated was because of the time component. And that's one of the things that's, again, salient within DIPG, and as an edge case provides a lens that's actually true across all cancers. As Sabina mentioned, time is the variable around which you have to optimize, right? Time is the variable which you have to actually make all decisions against. And that's what the original moonshot actually did, right? And that's what we as a community are trying to do. And so these consortia began committing to practices of near real-time data exchange and access, limited if no embargo of access to data, no limitation on who could access and use these data sets. And more importantly, the consortia recognized that even as consortia, we couldn't do this alone, that we had to find ways to partner with industry partners, as you'll see here, as well as more tightly link and look for ways to partner with the NIH and NCI, as you'll hear from Malcolm. The other lesson when you think about connecting is that you can't just connect across like-minded investigators and clinicians who are focused on pediatric brain tumors or even the DIPG. You have to really think about the biological context and opportunities to connect much more broadly, harnessing technological advancements that allow these platforms to make discoveries on behalf of investigators and enlarge your armamentarium and investigators and thought leaders so that you can get to that 400,000 investigator level. And one of the ways that I think we have been trying to leverage this is to look for opportunities where different diseases intersect. And these are, I think, really tremendous opportunities to think about. DIPG is a tremendous example of this because 1/5 of all DIPG patients actually share the very same mutation that is causative for another very rare disease that has nothing to do with cancer. Those two communities never actually interconnected with each other until that mutation was actually defined. So now a very rare disease in bone deposition called fibrodysplasia ossifica progressiva, or FOP, right, has a partner in crime with the DIPG community because both of them are trying to find drugs on behalf of a rare disease context. DIPG also launched new types of understandings around the way cancer is formed by identifying unique mutations in histones that were at least initially thought to be unique in the context of DIPG, but now we find that they also are shared in some context in leukemias, or in other bone cancers, or in adult head and neck cancers that also have histone mutations. So you find threads of connectivity across diseases that provide new opportunities for collaborative interactions and advocacy on behalf of the same mission. It turns out that you can even go further beyond cancer, and you can start looking at congenital birth defects. It turns out that if you have a congenital birth defect, it increases the probability that the biology could intersect with cancer. And it doesn't mean that if you have a congenital birth defect, you will get cancer, just like it doesn't mean that if you're a boy or a girl, you'll get cancer, even though it turns out that if you're a girl, you're protected to some extent from cancer. But you can harness these biological connections across very different diseases through the opportunities of data. There are some conditions that actually protect you against cancer, like Down syndrome. And again, these are communities of knowledge that can actually come together and bridge new opportunities for discovery. So for me, traveling to Australia for 36 hours and coming back is, to some people, sounds aberrant. But I have no doubt that every one of you in this room, right, would travel to the ends of the earth, right, over and over, right, on behalf of advancing the cause that we're here today for. The weird part about coming back from Australia is that you leave Australia, for example, I left on Wednesday at 10 p.m., and you land in San Francisco on Wednesday at 4 p.m., right? So you come back, and you actually travel in time, and you make up, right, for lost time. Now, unfortunately, we can't travel in time, right? But again, for me, every one of these trips highlights that time is the variable that we actually have to optimize against. And the only way to do that is through harnessing community, right? What you guys are doing here today, right? Advocacy on behalf of shared mission, shared messaging, and shared interests as a group, and on behalf of those other entities that could be your partners. And so for us, that is the main message across almost every element of our efforts, is to find opportunities for intersections. Because only then can we reach the moonshot level of efforts that are required to actually achieve the mission. So I look forward to partnering with all of you as often as possible in Australia or anywhere else. And I think this effort today highlights, right, the saliency of disease like DIPG, right, where time is the one variable we can't control that we have. Thanks.
Next we have Dr. Charles Keller with the Children's Cancer Therapy Development Institute.
Thank you, Janet, and good morning. Can everyone hear me okay? Okay, so speak louder. Okay, so I'll start out by thanking the congressional staffers and the congressmen and congresswomen they work with for considering this resolution that advocates for their constituents who have no public voice because of their age. The topic today is DIPG, diffuse intrinsic pontine glioma, and my assignment is to provide objective information about the preclinical gap, that's the rate-limiting step between the uniform mortality of this disease and creating tangible, science-justified, hope-filled new treatments. So first I'll introduce myself. So I'm a classically trained pediatric hematologist, oncologist, continuously funded by the National Cancer Institute for the last 15 years, beginning with my NCI mentored physician scientist training in the laboratory of Nobel laureate Mario Capecchi, and my current role is to be the scientific director of the 501c3 nonprofit biotech for childhood cancer therapy development called CCTDI. So I recently also co-founded a sister for-profit public benefit corporation, Biopharma, Artisan Biopharma, to partner in getting the discoveries to clinic. I'm sometimes funded by the pharmaceutical industry, but to be honest, the most valuable interactions that I have are the non-monetary joint ventures that we have with pharma. And so you can see that I'm a believer in the Bayh-Dole Act, whose spirit is to say that invention should move towards commercialization to make effective treatments available to the patients who need them. Now, for childhood cancer, the potential for giving back childhood in the form of durable cures with minimal side effects has never been better. And the humanitarian incentive is high, but much as everyone knows here, particularly the families, we just aren't there yet. In fact, we're kind of at a plateau. Every tool of science and engineering that we might dream of is at our fingertips. But while for adult cancer, there are 12 drugs so effective at extending and saving the life of an adult with cancer that are FDA approved every year. For children, there have been 8 since 1978. So that's something to think about. 12 every year for adults, 8 for children in 42 years. Of course, childhood cancer is rare, and depending on where you have the age cutoff, it represents about 11,000 patients out of the 1.8 million adults with cancer in the US every year. So the market incentive is low, but the government can and has begun to even the scales. I'd like to talk a little bit about that. Yet, historically, if FDA approvals are to be a measure of progress in pediatric brain tumors, then the 2 most recent brain tumor approvals, and these were secondary approvals, add-on approvals, were for CCNU, a derivative of mustard gas in 1976, and everolimus for a very rare form of inherited brain tumor called a subependymal giant cell astrocytoma, SEGA tumors. So no primary drug approvals have existed for brain tumors since the beginning of time. And we want to change that, of course. And the solution is multidisciplinary research, to be sure. So Congress has already created some tools to make this happen. I'll outline them. Certainly now, awareness is the key, making HRES 114 very timely. So first, allow me to describe the process of research and the preclinical gap. So curing cancer should be as easy as 1, 2, 3, right? First, you should begin to understand the cancer, identify a gene or a protein that would be your target of a drug. The second step is that you should test that particular drug for its rigorousness and effectiveness repeatedly in multiple different children's cancers in animal models. And third, you would move those drugs to national clinical trials. Now, I'll say out of those 3 steps, the NIH and the NCI fund the first and second step. But the first and third step, that's the discovery and the clinical trials. The second step is the notorious preclinical gap. So Malcolm and I share an affinity for the preclinical gap. So even though it's a gap, I kind of have an exciting precedent to share. So with private foundations, our laboratory organized 12 other labs worldwide to validate basic science and preclinical utility of a drug called panabinostat for DIPG. This work was published in the journal Nature Medicine in May 2015, and it was featured in the NIH Director's blog. And we certainly appreciate Dr. Collins' attention to our work, but we just wish that the example was less noteworthy and would be one instance in which a group of academic, community, and pharma members would move from a discovery to a reliable drug to putting a drug in clinical trials. But here's why I'm optimistic for the future. So Congress has created a series of carrots and sticks that facilitate pharma academic collaboration. So the Pediatric Priority Review Voucher, this is a really important legislation. It rewards drug companies that invest heavily in research and development of rare childhood diseases like cancer, and they receive a voucher for accelerated review of their next drug. This has a monetary value, which currently is about $90 million. Now, on the other hand, the RACE Act creates an obligation for pharmaceutical companies to find and test any potential adult cancer drug and test it in children. So both of these already enacted legislations came about from community awareness, very specifically, community action, but also legislative advocacy, such as is happening today. So the clear reason for cross-disciplinary collaboration exists, and now we just need to get the word out. So HRES 114 is a straightforward mechanism to stimulate conversations, particularly with pharmaceutical companies, innovation, and potential cures. So I'm very hopeful. So with that, I'll thank you for your attention and the advocacy of children who are touched by DIPG and other cancers, including my young friends Jack, Andrew, Kay, Philomena, and my friend Josh. Thank you.
Thank you, Dr. Keller. And now we have Senior Vice President of Oncoceutics, Josh Allen.
So let me begin by thanking the patients in the room and the families that are gathered here today to represent the patients who couldn't be here or are no longer with us. You're really the reason why we're all gathered in this room from many corners of the globe, as you've heard. And you're the reason why we're fighting this disease every day. So thank you for that. So my name is Josh Allen. I'm Senior Vice President of Research and Development at Oncoceutics. I'm also the son of a childhood cancer survivor, and I discovered ONC201, a new drug that is being developed by our company for the treatment of DIPG and other forms of lethal brain cancer. As many of you know, the mortality rate in the United States due to cancer has been declining in general, thanks to the tireless efforts of the oncology community that have revolutionized the management of many forms of cancer. Some of these cancers I was taught only 10 years ago in school to be incurable forms of this disease are now sent into remission by breakthrough therapies. And unfortunately, DIPG is not one of these. The breakthrough progress that we've witnessed in some forms of cancer is only possible when the entire community is working together. This includes patients, their advocates, physicians, academic researchers, regulators, and industry, among others. For DIPG, the academic researchers, as you've heard, have been hard at work making exciting discoveries that help us understand this disease and how to best fight it. The physicians are passionate and highly motivated to do whatever they can do to help their patients. The patients, their families, and their advocates are among the most resilient and inspiring people you will ever have the privilege to meet. And that leaves industry and regulators, which I'm here to discuss. Oncoceutics is among a very small number of companies that have included DIPG in its development focus of novel therapies. There are myriad reasons why this group of industry members is so small. First, clinical trials for drugs to treat brain tumors in general have a very high failure rate. DIPG is even worse. This is a form of brain cancer that has seen decades of clinical trials that across the board have failed to move the needle on outcomes. Second, DIPG is considered an extremely rare disease, as touched on earlier by other speakers, having an annual incidence of 200 to 400 patients in the United States. This makes it hard to find patients for clinical trials. It also makes it hard to finance because the development costs can't be recouped in the event of commercial success for such a rare disease. As a result, DIPG has not been a priority for the drug development industry. And this has to change. We can't continue to let hundreds of people, many of whom are children, have their lives stolen by DIPG every year. The good news is that there's cause for hope. Grant-funded research has breathed new hope into the community. We've only recently begun to understand exactly what goes wrong with DIPG in the first place and continues to drive its growth and spread. This allows us to understand why so many prior therapeutic efforts have failed and cast light upon a series of ways in which DIPG initiates and drives and we can potentially use for attack. That gives us hope that new therapies could be targeted at those kind of defects to finally make progress. Not only is there cause for hope, but there are things that people in this room can do to help. In the case of Oncoceutics, we would not have been able to develop ONC201 for DIPG without government-supported research. I discovered ONC201 as a researcher at the University of Pennsylvania, supported by NIH and foundation funding. In addition, Oncoceutics has been supported by direct grants from the National Cancer Institute that have enabled us to run clinical trials. Indeed, almost every step of the ONC201 discovery and development process has been critically enabled by NIH and other government agency grants. Without this support, we simply would not have been able to discover the drug. We would not have been able to bring that drug to clinical trials to patients. We would not have been able to run brain tumor trials. And finally, we would not have found a mutation that is common in DIPG and other kinds of brain cancer that is key to making these tumors respond to that agent. We now know that the drug really helps some of these patients by shrinking their tumors and improving their symptoms. Years ago, without this data, the idea of funding clinical trials for a new drug that works in a new way for a disease like DIPG, where nothing has ever helped in the past, was quite simply a no-go for private investment. Grants such as the NCI SBIR program and many brain tumor foundations represented in this room stepped in and critically enabled research and clinical trials of this drug. We are eternally grateful for the support by this community that has enabled us to discover and develop ONC201. But how many other promising novel therapies discovered in academics' labs never had the chance to be tested in clinical trials? And how many more worthy therapeutic concepts are we going to read about in research journals that will never make it out of the lab and reach patients? I call upon you to increase support for federally funded basic, translational, and clinical research for DIPG as suggested by House Resolution 114. This would accelerate research and clinical trials to find new treatments for the most deadly form of childhood cancer. It would expedite the path to approval for investigational therapies like ONC201 that are showing promise in clinical trials. These efforts will be critical in allowing regulators and more industry partners to join the rest of the DIPG community in doing everything that we can to help these patients live longer and better lives. Let's give these patients, their families, and the community the help and hope that they deserve. Thank you.
And now we have Dr. Malcolm Smith from NIH.
I'd like to thank Janet Demeter and all of the hosts today for allowing us to present at this meeting. All the families and organizations who have come together to put a spotlight on this issue, to emphasize the need for continued research, and to honor the children we have lost to DIPG and their families and those facing a diagnosis today. And thank you as well to Congresswoman Spear for her leadership of the Childhood Cancer Caucus, as well as the Biomedical Research Caucus. And thank you to her and her colleagues for their continued strong bipartisan support for NCI, for NIH, and for childhood cancer research. And thank you for this opportunity to speak today about research supported by the National Cancer Institute and across the National Institutes of Health to advance progress for children with DIPG. As many of you know, NIH is the nation's biomedical research agency, and NCI is one of 27 institutes and centers that make up NIH. NCI and all of the NIH institutes are primary supporters of basic biomedical research, and both NCI and the National Institutes for Neurological Disorders and Stroke, or NINDS, support research on brain tumors. My role at NCI involves overseeing a portfolio of large translational and clinical research programs that are funded by NCI. That includes the Children's Oncology Group, the Pediatric Early Phase Clinical Trials Network, the Pediatric Preclinical Testing Consortium, and a program that I'll talk more about today in detail, the Pediatric Brain Tumor Consortium, or PBTC. I'll focus on two major areas of research. First, the basic science studies that allow us to better understand the biology of DIPG and other pediatric brain tumors. and make discoveries that we hope will establish a foundation for future clinical progress. And second, I'll describe the clinical research programs to evaluate new therapies and approaches that are needed for children with DIPG. I'll share examples from NCI, from NINDS, and from other areas of NIH in the next 5 minutes, so I'll be talking fast to try to get through as much as I can. First, and I'll be talking about the things that are shown on the slide collage up that you see. NCI has provided long-standing support to Dr. Suzanne Baker, and you see her in the bottom left picture. To Dr. Baker and her outstanding team at St. Jude Children's Research Hospital. Theirs was the first group to report on the histone mutation that's now taken as characteristic, a basic characteristic of DIPG. And this team is conducting basic and translational studies in an effort to develop effective, relatively non-toxic therapies through a better understanding of the biology of DIPG. I'd also like to highlight Dr. Michelle Manji's work, the picture in the upper right. She's at Stanford, and she's been supported throughout her career by NINDS, and more recently received the NIH Director's Pioneer Award through the NIH Common Fund. The Common Fund is an important way for NIH to support research that transcends what any single institute can support, and it's also the home to the Gabriella Miller Kids First Research Program, of which NCI is a key collaborator. If you're familiar with Dr. Manji and her work, you know that she's one of the foremost scientists studying and developing new therapies for pediatric brain tumors like DIPG. Her basic and translational research, supported by NIH and many pediatric brain tumor foundations, has led to NCI-supported clinical trials, both current and in development. I also want to call your attention to Dr. David Alice. He's in a picture in the upper left corner. Dr. Alice at Rockefeller University. NIH support to basic researchers like Dr. Alice has been critical to advancing our understanding of the biology of DIPG. Dr. Alice's research for 30 years has been on histones, a protein that helps package DNA. It wasn't until histone mutations were discovered as central to DIPG in 2012 that the connection between Dr. Alice's many years of research in DIPG was made. And now, Dr. Alice has become a passionate childhood cancer researcher with a large NCI grant supporting important studies on the role of histone H3 and how it drives the development of childhood cancers like DIPG. Additionally, both NCI and NINDS are funding young investigators, and this is critical because these are the investigators that will be making the discoveries of tomorrow. Support grants like the NINDS grant to Dr. Sharam Vanetti in the lower right, who's at the University of Michigan. He's building upon what we already know about histones and building on the work that Dr. Manji and Baker and Alice have done to better understand how these histone mutations rewire the cellular circuitry to make DIP such a terrible tumor. Additionally, through the Cancer Moonshot, NCI is supporting a team of Dr. Resnick's colleagues at CHOP to create a pediatric tumor cell atlas. This is an incredibly in-depth cell map at the molecular level to understand the tumor cells, the tumor microenvironment, and the immune system. The CHOP team for the Pediatric Tumor Atlas is studying 3 high-risk cancers that together account for more than 50% of all childhood cancer deaths. One of these cancers is childhood high-risk glioma, including tumors with the H3 histone mutation. And so the Atlas findings will be highly relevant to DIPG. We are also working across NIH and across the childhood cancer community to try to ensure that promising basic science is translated as quickly as possible to children in the clinic. As an example, NCI and another NIH center known as NCATS, or the National Center for Advancing Translational Sciences, partnered with Dr. Manji, with Dr. Kathy Warren, now at Dana-Farber, previously at NCI in our intramural pediatric oncology branch program. NCAS partnered with Dr. Warren and Dr. Manji to use preclinical information to improve clinical approaches. They had a high throughput screening in cell culture of 2,700 compounds on several DIPG cell lines, and their results identified several combinations of interest and potential importance for future clinical evaluations. NCI is also expanding the Pediatric Brain Tumor Consortium this year with increased funding. This will allow the PBTC to add up to 6 additional sites, and it will also allow the PBTC to increase its organizational capabilities so that it can develop more clinical trials and have them available for children. An important part of this expansion will be the ability to do more for DIPG. And one example being a clinical trial that Dr. Manji is leading, evaluating a drug that inhibits a protein called ADAM10 that's involved in the growth of glioma cells, including DIPG cells. The expansion of the PBTC will allow it to expand its clinical research programs for DIPG and will make its clinical trials more accessible to families and their children across the country. In addition to the PBTC, which focuses on early phase clinical trials, NCI, in collaboration with Oncoceutics, as Dr. Allen described, will soon be launching a trial through NCI's National Clinical Trials Network. It's a partnership between the NCI-supported Children's Oncology Group and one of NCI's adult clinical trial groups to evaluate ONC201 plus radiation for children and adults with newly diagnosed gliomas, DIPG, or other brain tumors with the H3K27 mutation. As you heard from Dr. Allen, NCI's Small Business Innovation Research Program played an important part in supporting the development of ONC201, and this phase 2 trial supported through the NCI National Clinical Trials Network will build on that progress. So I know I've gone over my time and I've not done justice to all of the promising research that we could have talked about today, but want to leave time so that you can hear from David and Jace. I thank you again to Janet and all the families and organizations represented here. For those who have lost a child to DIPG, I know that the discoveries we're supporting today are coming too late. Please know that my colleagues and I within NIH and throughout the country understand the urgency for all children with cancer, but particularly for children with DIPG. And we look forward to continuing to collaborate with you to keep pushing this field forward as fast as we can. Thank you.
And now I'm especially honored to introduce David Ahrens, the CEO of the National Brain Tumor Society. This is our hero in philanthropy. I have a small little brain tumor foundation in Agua Dulce, California called Jack's Angels, and I'm just thrilled to have my hero here. Thank you.
Thank you, Janet. And of course, the real heroes are patients and families, and we honor them today. Thank you so much, Janet, for putting this whole thing together. Thank you to Feed DIPG, Smashing Walnuts for collaborating to help. Thank you to Congresswoman Spear, the staff, all the congressional staff and members that took time out today to be here. It's really, we're grateful that you're here, grateful that you're listening. Special shout out to Sandy and Richard Perkins, great advocates and volunteers with National Brain Tumor Society that traveled from Arizona, especially to be here, and to Beth Ann, who's here as well. Thank you all. We have a lot to do, and we're hearing a lot about promising projects and things that are going on that are promising. But as you know, we have a lot of hope. They give us a lot of hope, but hope is not a strategy in and of itself. But what you're hearing is science has now emerged to form a strategy. We didn't have a strategy 20 years ago. Now we do. And so that's what we're all talking about, is that hope is turned into a strategy that's actionable, but it's dependent upon Congress and staff and members taking action by by passing this resolution and passing policies, passing appropriations bills to give us the fuel to get it done. This is a terrible disease. This is a disease that tears at the soul. Parents and children who are facing this disease are almost always met with treatments that don't work. They're desperate. They scour the world to find answers. We get calls from parents all over the place that have quit their jobs, raided their 401k plans to pay for treatments, to pay for the travel. This is a disease that is not just about health, which is bad enough, but it creates real financial and economic hardships. It tears away the soul in lots of ways. If there were a stronger word than urgent, it should be applied to finding a cure to DIPG. Dr. Resnick said time is a key variable. Well, that time is right now to act. That's our variable is right now. We have to do much more. But overall, we need an attack plan against DIPG and other pediatric brain tumors that takes on multiple strategies: scientific, healthcare, awareness, and advocacy, what we're here to do today. Scientifically, we need to make more progress to understand DIPG and do the things that Dr. Keller talked about, identify vulnerabilities and find more promising treatment development approaches. We need better health care. We need to develop those treatments with state-of-the-art cancer centers like the researchers going on with Dr. Mueller, Dr. Packer, Dr. Resnick, and others. We need to enact policies that make sure that DIPG patients have access to the state-of-the-art molecular diagnostics. And today we call upon policymakers, insurance providers, and everyone to make sure that DIPG families and patients have access to the kind of state-of-the-art molecular profiling necessary to even understand and diagnose their tumor and put them in a position to get the right therapies at the right time. Not all patients have access to covered molecular profiling. That needs to get done. Financially, DIPG families face terrible financial toxicity, high medical bills, astronomical costs. We have to do better by that. And awareness and advocacy. We need to raise international and national consciousness of this so that children and their families are not alone, and all those who want to organize to do something about us can come under this big tent that Janet has helped create today. National Brain Tumor Society we're part of this movement. Our overall strategy to defeat brain tumors and conquer and cure them once and for all is defeat, connect, and change. And we funded, at the beginning of our journey, those studies that Dr. Smith talked about, the funding of Cynthia Hawkins and Dr. Baker to discover those histone mutations. We've brought together industry, drug companies, FDA, to try to deal with drug availability in clinical trials so that talented clinical trialists like Sabine Mueller have the opportunity to get the drugs from the companies to evaluate them. During the last 3 years, we put together a team science project, knowing that one lab alone is not going to win the day, but it's going to take all of us working together. So we have a team of St. Jude's, German Cancer Center, Montreal McGill, going hard at it under defeat brain tumors with support from NBTS, St. Baldrick's Foundation, Raleigh Foundation, students supporting brain tumor research, and others to go develop those models and develop liquid biopsy approaches and find new drugs. Going forward, we're going to be funding promising DIPG, pediatric high-grade glioma research stemming from this project. We're very pleased to be supporting and working with Oncoceutics to try to make the most out of ONC201. On the advocacy front, NBTS, together with other groups, is focusing like a laser on increasing federal support for DIPG research. One of the areas where we focused over the last few years is with Defense Department funding. Yes, the Defense Department funding DIPG research. And since 2010, the Defense Department, through one program alone, the Peer Review Cancer Research Program, has provided over $6.1 million in specific DIPG research. But that has to be re-upped every year. It depends on us showing up, making noise, and demanding that the Defense Department get the funding from Congress to do it. An example, just 2 years ago, Dr. Eric Rabb, right up the street at Johns Hopkins, received a $650,000 grant to study DIPG. Another one in 2017 by Juan Fueyo at MD Anderson is now trying out the common cold virus on DIPG. which has been now in a phase 2 trial in adult glioblastoma, original funding from NBTS. And so we're trying multiple approaches here with viruses, but a lot's going on. Thanks to the work of advocates in Congress coming together, the National Institutes of Health has increased their overall pediatric cancer funding from $342 million to $595 million from 2015 to expected 2020 funding. And one example of DIPG research now underway, thanks to that advocacy that we so need, is the work going on at University of Michigan with Dr. Vanetti, $450,000 to look at how we can alter tumor metabolism as a strategy. How can we basically starve a brain tumor by changing the nutrient uptake? That's the strategy, and we're following that. We're following the science, and the NIH is doing that. Project Hope Care is another example of the NIH committing under the NCI $2 million to do single-cell sequencing of pediatric high-grade gliomas, including DIPG, just a year and a half ago under Dr. Sharpless. Gets a lot of credit for that. At the same time as we need to develop the science, we have to unite the community and get these clinical trials further and faster. ONC201 that you've heard about is a great example that Dr. Allen is leading. And there's some other terrific examples. Cincinnati Children exploring an important drug in combination with radiation. Baylor exploring CAR T therapy. You've probably heard about CAR T in the news of it's an immunotherapy approach. Baylor, as well as Michelle Mangi's work at Stanford exploring this. Mark Atlas, a researcher looking for how to take an old drug like mebendazole, which was used to treat infections of intestinal parasites, and apply it in a DIPG setting. So we're seeing creativity in science to take the old and apply it in the new. And Dr. Mueller's innovative work on enhanced delivery using irinotecan liposome injection, and her trial evaluating fimipinostat, a MEK inhibitor from Curis. Different approaches, Dr. Mueller taking different shots on goal to help DIPG patients. Of course, the multi-arm study by Oncoceutics, and one we have to mention, Dr. Tinkle at St. Jude's, who's looking at a blood-brain barrier penetrant inhibitor, but looking at how to use that to evaluate in combination therapy with radiation. So a lot going on. Congress and patient advocacy working together in an urgent and collective matter can get big things done. Together, we got the Childhood Cancer STAR Act passed just 2 years ago, providing $150 million over the next 5 years in biospecimen collection, survivorship studies, cancer registries. This is an example of what we can do when we work all together. Bipartisan legislation signed into law in this administration, focal point on childhood cancer. Together, we can make a difference. Working with groups like Defeat DIPG, DIPG Collaborative, Dragon Masters, Jack's Angels, Max Cure. There's so many groups who are all eager to work together and do it. With improving science, access to health care, and bold advocacy, and the partnership of the great congressional staff here in this room, which we thank you, we can do much bigger things. We can make progress. Thank you so much. We urge Congress to pass the DIPG resolution and elevate DIPG on the congressional priority list. Thanks again for all those attended today. Let's feel the passion and turn that into results. Thank you.
Thank you so much, David, and thank you to staff. Now we have Jace Ward, and as I said, he's a pre-law student at Kansas State. I just wanted to give a special bit of thanks to him. This event is, to a certain extent, to his credit and his mother. I learned of their case over the summer due to an article that Amanda Haddock with Dragon Master Foundation, she had written about him and the struggle to keep the ONC201 trial alive. It was, there was a threat, it would have been shut down because of inadequate funding. And thanks to the community collaborating, now the trial is open and kids are able to benefit from it. At any rate, thank you so much, Jace, for being with us. The floor is yours.
Before I get started, I'd like to... Before I get started, I'd like to take a photo with you guys just to show all of the kids who weren't able to make it here that they're not fighting alone. They have help, and we're here. So. All right. We're good to go. Thank you. Good morning. I am one of the many faces of DIPG. On May 17th this year, I learned what a diffuse intrinsic pontine glioma is. As I learned, my vision blurred and everything went numb. But my doctor said something along the lines of an aggressive, inoperable, terminal tumor in the center of my brain. The only other thing I learned that day is that I had an estimated 9 months to live. I would die one day before my 21st birthday. There are no words to correctly explain my thoughts in the following months. I can only tell you that brain surgery doesn't hurt, it'll just make you forget how to walk, and radiation masks are straight out of a nightmare. If you're doing the math, then yes, I have about 4 days to live. In 4 days, I will consider myself lucky to be alive. I can't promise I'll be back here next year, which is exactly why I respectfully ask you to co-sponsor House Resolution 114 before you leave this weekend. The rush is, DIPG won't wait until this is convenient. DIPG won't wait until we are ready. While we have been waiting to take a solid stand on DIPG, DIPG has been taking the sight, the hearing, the speech, the ability to swallow, and eventually the breath of thousands of kids across this country. Cruelest of all, we will always be fully aware of the torture that we will one day endure by DIPG, as DIPG does not affect our cognitive ability. You hear my urgency, so once again I plead, please sponsor House Resolution 114. Let's celebrate National DIPG Awareness Day on May 17th this year, one year from my diagnosis. Since May, I've been telling everybody, I can't die, I'm busy. It's true, but the real truth is, at almost 21, I'm just not ready to face the progression of DIPG. And at an average age of 8 years old, how could anyone prepare for this? Most nights, I lay awake wondering what all those children on this path before me were thinking. I wonder if the older ones ever wondered why despite their parents' prayers and pleas, they could not be saved. And I wonder if the young ones even knew why they were at the doctors. The point is, we just don't know. Because as a nation, we are not aware. We have failed to hear these children and their families. Today, if I told you there's a person walking around who is killing 300 to 400 kids every single year, I would imagine we would all go home, hug our families, and turn on the news to watch our nation save the day. Well, I have gone home, and I have hugged my family. DIPG is not rare. DIPG is just rarely talked about. I can't expect you to fix something that you don't know about. So in the US, pediatric cancer kills more children than any other disease annually. Of those deaths, more than 15% are from DIPG. We need this entire country to be aware of that kind of killer. House Resolution 114 is an important step in demanding attention be paid to DIPG when funding public or private research. Awareness will lead to more funding and will lead to more cures. We can solve immediate life-threatening issues with simple awareness. In my state of Kansas, at the largest research hospital, the director of oncology believes that there are only 40 cases of DIPG in the US per year. No, sir, but I can tell you there are at least 7 active cases in Kansas. Jessica, in Wichita, Kansas, was diagnosed in July soon after she graduated high school. She did all her doctor recommended, which was steroids and radiation. There was no discussion of clinical trials or a biopsy to discuss mutations. In 6 months, Jessica became wheelchair-bound, has double vision, cannot use her hands reliably, and blood clots are forming in her chest. Steroids have ran their course, causing disfigurement and weight gain. Jessica watched one of my interviews on local TV in December, where she first learned of the drug I am taking. Unfortunately, it is too late for Jessica to start the medicine until she waits for her tumor to progress further. My child may not have helped Jessica, we'll never know, but she deserved to know her options and participate in the discussion. We as humans have failed Jessica. By the grace of God and the people like those in this room, my mother was able to find a different course of action for me. If you want me to be really honest with you, my hope came from a warrior-hearted group of Facebook moms. That's right, Facebook, I know. These moms share their children's experiences with trials and stay up to date with the research. In fact, their experiences are the most up-to-date data that we have on most of these trials. These parents rallied for me to receive expanded access on ONC201 when I was too old for the clinical trial and access was denied. They then funded the expanded access with fundraisers for 86 kids last year. They were aware that ONC201 is the best chance I have for my mutation currently. They were aware that 70% of those with DIPG have the exact same driving mutation and was responding to a new drug called ONC201. Because these selfless, grieving parents were aware, I am very fortunate. I now take the drug 5 pills 1 time a week without many side effects. Maybe it's the radiation that currently keeps me symptom-free and my tumor will come back with a vengeance soon. Maybe it's the drug and I've got a year or 2 or more. It's just too early to know. That's more time for me to enjoy school, accomplish my goals, hug my friends, and love my family. Maybe another birthday, or if I'm really lucky, I could graduate college. But I am one of the very few examples of awareness. I wasn't supposed to be here today. Awareness 3 years ago caused a neurosurgeon to lead the way to safer biopsies, which gave researchers a way to detect tumor mutations, which allowed a new drug company to confirm a brand new type of drug could combat 70% of tumors just within the last year. Well, my mom, finding a few aware parents who knew more than their doctors about choosing our best course of action from Facebook, is the only reason that I'm here today. My story looks different today because parents who have lost children pleaded with my mother to make awareness her priority because we cannot depend on awareness within the medical system. As a nation, we cannot allow ourselves to continue being complacent with our lack of knowledge and awareness. Our inability to prioritize DIPG patients have resulted in death after death. Just as previously incurable diseases have shown time and time again, awareness is a catalyst for a cure. Please give us 1 day a year to use the biggest forums we can find to tell people about DIPG and trade for all of the years of lives lost to this horrible drug or horrible tumor. We'll use the day wisely to inform the public, guide patients to new treatments, doctors to Center of Excellence, and researchers to data. Earlier, I said that I could not expect you to fix something you don't know about. Well, now you do know, and my life is on the line. No pressure, and good luck.
Because we're limited in time, we're going to skip the Q&A. You're welcome to write down questions to the particular panelists you'd like to ask, and our volunteers on the outside have a basket for you to leave those in before you leave the briefing. Next, we're going to hear from some parent advocates who have a very, very powerful message.
Hello, everyone. Can you hear in the back? Thank you so much, Janet, for everything that you've done to put together this event, and to the whole DIPG advocacy group, as well as to Congresswoman Spears' office for supporting this event. It's hard to go after Jace, because I'm not sure there is any more compelling case I've heard than what he just put forth to you, and I, for one, am blown away by his poise and strength. My name is Jenny Mosier, and I'm the Executive Director of Michael Mosier Defeat DIPG Foundation. In September of 2014, we learned that our son Michael had DIPG. It was his second week of kindergarten and a week after his 6th birthday. In a flash, our lives were shattered. Doctors told us we could try experimental drugs, but he would likely die within a year. We wanted hope, so we told Michael that radiation, needle pokes, and medications that made him sick were all to help him get better. He did everything that we asked, but as expected, nothing worked. Just weeks after diagnosis, Michael couldn't walk. He took steroids that inflicted horrific side effects, including massive, rapid weight gain. He had double vision. His body became paralyzed, bit by bit, starting on his left side and working its way to his right until it took over his entire body. DIPG even stole his smile. On May 17th, 2015, just 8.5 months after diagnosis, Michael passed away. The first time our family ever heard the term DIPG was when Michael was diagnosed. We knew that we needed to increase public recognition of DIPG, so we started in our community. In 2016, Governor Hogan established May 17th as the first ever DIPG Awareness Day in Maryland. Soon thereafter, I heard from multiple families across the country who wanted to establish DIPG Awareness Day in their state. Other states, including California, Michigan, and Pennsylvania, had also recently had DIPG awareness recognitions. And the DIPG community realized that working together and coalescing around a single date of recognition would amplify our efforts to make the most impact. And so, the DIPG Across the Map initiative began to establish May 17th as DIPG Awareness Day on behalf of all of those affected by DIPG in all states. In 2019, thanks to the work of many foundations and individuals across the country, 32 states recognized May 17th as DIPG Awareness Day. We expect more this year as we continue this companion effort to the work that we are doing on the federal level. The House of Representatives has the opportunity to stand with thousands of families across the country who have lost children to DIPG or are fighting right now for the lives of their kids. The burden of fighting for a cure should not fall only on the shoulders of those families who have already paid the ultimate price through the loss of their loved one. HRES 114 alone cannot stop this disease. But recognition by our federal government matters. By raising awareness of this disease, we will build the coalition of supporters who are rallying for our children. Official government recognition of the need for attention and funding for this disease is meaningful as we all work to increase the resources available for the researchers who are fighting so hard for a cure. As DIPG tumors grow, they literally take away the ability for a child to speak. We ask the House to be a voice for our kids through HRES 114 by recognizing the importance of their lives and calling on all Americans to support the efforts to find a cure. Thank you.
Thank you so much, Jenny. I followed Michael's story, and that's the reason that our national resolution has the May 17th date, because we were already showing kind of a consolidation around that day, and we wanted it all to reflect it in our efforts together. And now I want to welcome Elizabeth Bassar with the Julia Barber Foundation from Knoxville, Tennessee. She's my partner in crime with the DIPG Advocacy Group.
Hi, Hi, thank you so much for being here. My name is Elizabeth Pizarre. I am with the Julia Barber Foundation. This is my son, William. He is probably the littlest member of our DIPG advocacy group, and he comes along with us. My daughter, Julia, we have a picture of her. She actually passed away May 17th of 2016. Which got me involved in this whole legislative process. I knew what DIPG was. She was diagnosed in 2015. She was sliding down a slide ladder. Her balance was off, and I didn't realize that she had a brain tumor. I thought, she had an inner ear infection. It never occurred to me that she had a brain tumor. I am a juvenile law attorney. I've spent 12 years representing kids that are neglected and abused. And I never thought that my daughter had a brain tumor. And so she went to the pediatrician. I went to court thinking there's no big deal. That was Monday. Tuesday, we go to Children's Hospital, and we're told my daughter's going to die, and there's nothing I can do to save her. There's nowhere I can take her. In the whole wide world, there is no way that I can save my daughter. So I was so upset, obviously, because there's nothing that we could do. But I was also so upset that I never thought brain tumor, because nobody talks about it. It's this big pink elephant in the room that is killing our children, but nobody talks about it. It's like it's too scary, so we just ignore it and hope it goes away. That's not acceptable. That's not okay. And that's why we are here with the DIPG Advocacy Group. We formed the DIPG Advocacy Group 3 years ago. 2017, so a year after Julia died, we formed the DIPG Advocacy Group in a way to have all of us together as a group to make our legislative bodies care about our kids, because our kids are important. And it's not just DIPG kids, it's all kids that have pediatric cancers, it's kids that have brain tumors, people need to care about our kids, they're important. And it's important that we talk about it, because just in my world, our foundation is in Knoxville. I've had people come by. There was a lady that came by. She started crying. She saw my booth. She said that her 14-year-old son died from a brain tumor. He had headaches for a couple weeks. She didn't think anything of it, and he died in his sleep. And it was an operable brain tumor. It wasn't DIPG. He could have sought some form of treatment. She didn't know, and that's just our world. Recently, I've had another friend whose nephew has been diagnosed with a brain tumor. Her sister took her son to pediatricians. They told him that he's 12, he just has allergies. That happened a couple times, but because of our foundation and what we're doing and the DIPG Advocacy Group and the work that we do, they took him to other doctors. And then they found out that he has a brain tumor, and now he's seeking treatment, and he didn't die in his sleep like the 14-year-old because they talked about it. And that's why we're here. That's why we have to talk about it. We have to make a difference, and that's why May 17th awareness is so important. House Res 114, we've been fighting for this resolution. We had House Res 69 before that, and we just need your support. We need you to help us reach out to your congressman and let them know that this is what you need to support. Staffers, we need your congressman on board. So please go ask them to co-sponsor. We have a lot of people here, but most likely we have a lot of people in your district, because this type of disease doesn't lend itself to being able to talk about it. I've talked about it a lot, but I almost started crying when I was talking about Julia, even though I talk about her a lot. It doesn't lend itself to an easy way to discuss it with people. So we would ask you to support it. We do have a Senate resolution also that we had last year for May 17, 2019. We're going to have a Senate resolution again for this year, May 17, 2020. It's a national day, but we also need it on the House side. So we need both parts of government to work together for this cause that is so important to everybody. So thank you so much.
Now the Morin family is going to come forward. Are you there? Jill.
Hi everybody, my name is Cam Morin. I'm here with my two rocks, my beautiful rocks, Jill and Phoebe. And we're here on behalf of our son Luke. Luke passed away on April 25th, 2018. And it's horrible to hear that kids get these diagnoses. And by the way, Jace, kudos. You're rocking it, and I'm so pulling for you. But you know, you hear 8, 9 months. It's awful. It's just awful. Luke died 17 days after he presented his first problem, which was, like a lot of these kids, trouble balancing, drooling, and just loss of motor control. The story was the same for us, and it was brutal. I stand here as an angry parent, not just a sad parent, an angry parent. And I look at you young kids, you staffers here in the front rows, and I hope that you can see the pain that I feel and can go back and say, listen, this is absolutely silly that we have House Res 114 sitting around and it's not done, it's not complete. So I ask you to go back and advocate for us and help all of these incredible people sitting here at the dais get the funding they need and the awareness that this requires to do something about it, so that other parents don't have to go through what so many of us in this room have done. And Jill, I'm sure you have something to say.
Phoebes, would you like to well, I'm Jill Morin, Luke's mom, and Cam's rock, I suppose. He's my rock as well, and Phoebe also. I will end by kind of just expressing that this is urgent, this cannot wait, a cure cannot wait. DIPG didn't wait for Luke. DIPG didn't wait for any of these kids. This is one day that we're asking for, and I'm proud to be standing here as part of this group. I'm proud to have worked with Janet and all of you wonderful people, part of this community. I'm proud to use Luke in our story as an example of the worst, which is DIPG, which is the worst of the worst. Brain tumors are the #1 cancer-related death of our children. DIPG is the worst of the worst. And our Luke is, I'm happy to represent Luke to be the worst of the worst. He was a wonderful little boy, articulate, energized, happy, healthy. He was it was about 72 hours of significant symptoms. We found out about the diagnosis, and he was gone 17 days later. So just take a minute, let that settle in, and just imagine if it was your dog, if it was your grandma, if it was anyone. My son, her brother. Thank you.
And now a surprise visit from Dr. Monte.
My name is Michelle Monge. I am a pediatric neuro-oncologist at Stanford University. My research laboratory is focused on understanding DIPG, how it arises in the developing brainstem of children and adolescents, what DIPG cancer cells depend on for growth, progression, and spread, and how we may best treat it. My clinical research program endeavors to bring what we discover in the lab to clinical trials of new therapeutic strategies for DIPG. Brain cancer is the leading cause of cancer-related death in children, and DIPG is the leading cause of brain cancer-related death. To date, DIPG is universally fatal, and the average survival is less than a year. It strikes approximately 400 children, adolescents, and young adults every year in the United States. And given the median age at which DIPG occurs and the average life expectancy in the United States, DIPG accounts for approximately 24,000 life years lost annually. I first became aware of this disease in medical school when I cared for a little girl with DIPG named Emily, from the day of her diagnosis until her death 6 months later. I was stricken by the unthinkable cruelty of this brain cancer and stunned by the horrifying realization that, at the time, we knew virtually nothing about DIPG, had no experimental model systems to study it, no knowledge of even the most basic aspects of what drives this seemingly intractable cancer. And, worst of all, no way to even slow down its relentless progression. In the nearly 2 decades since her death, motivated by Emily and the ever-growing list of children, adolescents, and young adults I've cared for and lost, I have been part of the small but growing team of doctors, scientists, and parents fighting to understand, treat, and one day cure this terrible childhood cancer. In that time, there's been enormous progress, as we've collaboratively developed tools to study DIPG in the lab, uncovered the molecular underpinnings sparking this malignancy, determined ways in which DIPG takes advantage of the rich, growth-enabling environment of the developing childhood brain, and exposed several vulnerabilities of DIPG that we are working to translate to new therapies. What we are discovering about DIPG has also taught us important lessons about other brain cancers, and has elucidated new possible therapies for more prevalent diseases like glioblastoma. This progress forward has come only from a collaborative effort that crosses disciplines and borders. It has been supported by those who have seen the disease through a variety of circumstances and could not turn away from this urgent unmet need. How much larger the effort would be, how much quicker we would find effective therapies if more people were aware, if more people and more resources joined us in this fight. Thank you for taking the time to consider this initiative to raise much-needed awareness that will benefit children, present and future, who face DIPG.
Thank you so much to our panelists. It's truly been an honor to have you with us. And to the congressional staff that are here. I recognize some of you. I started this journey with the DIPG Awareness Resolution in 2016. I've gone to many offices for many families who are, I'd like you to come forward now with your pictures of your kids. It's time. I've brought stories to many offices that I fear have fallen on deaf ears at times. This was an experiment in democracy because we have so few avenues of direct communication with our representatives, especially our representatives in the House. A lot of our reps don't have town halls. We just need to know, we just want you to know that with our advocacy efforts, we are representing real people, mostly dead, and we need to support those who are fighting for their lives right now. They need to know that we've got their back, that we care, and that we're not going to look the other way. I do not believe that we are the country that looks the other way. I believe in our democracy, and I believe that this is a unifying principle that could help heal, so much. Because when we come together about things we believe, like how we value our children, how we value life, it makes all those other decisions we have to make where we're so politically divided so much easier because we're together. So thank you to congressional staff for joining us today. It's an honor to have you. This presentation is for you. And again, thank you for taking the time to consider these very real people across the United States, across the world. Let's lead. Let's lead in the world by passing this. Thank you. You have the power to attach hope to brain cancer with a simple signature, a simple agreement to be resolved and united. Thank you so much for your consideration. And thank you all the families who have come. I love you.